Okay, this is the final preparation for lecture 3, and this part just focuses on kind of the steps, the basic mechanics of how the DNA replication process happens. And this happens every time cells need to divide. They have to copy the DNA so that a copy can go into each of the daughter cells from the original cell. DNA polymerases, I think I mentioned this last time, they add nucleotides to the 3' OH on a strand. And so we say that DNA grows 5' to 3'. And what that means is that for each nucleotide on the 3' end, that is where the new nucleotide is added. So there's a new nucleotide, and then it has a 3' hydroxyl, and then a new nucleotide is added to that, and so on. And so it builds in a particular direction. Okay, so that's just the conventional way, 5' to 3' synthesis. That's how we describe it. And on the leading strand, remember that an RNA little primer is required to initiate the synthesis, because DNA polymerases can only add nucleotides to an already existing 3' hydroxyl. So they can't just suddenly start making DNA without a previously existing strand to add on to. And so the RNA primer is made, and then the synthesis occurs. So that's 5' to 3', and then the nucleotides just get added, right, until you get to the replication fork. And then as replication proceeds, on the leading strand, the DNA just unwinds and opens more, and you can just keep going, keep adding. And so the leading strand is built relatively quickly because there's no sort of extra steps that are required. But in the case of the lagging strand, because of the directionality, an RNA primer is made 5' to 3' like this, and then the nucleotides are added. And then the DNA opens more, and then another primer is here, and then the nucleotides are added. And then it opens more, and there's another primer, and then the nucleotides are added. So the DNA, because of the direction, because the enzyme can only build DNA in one possible way, you get all these pieces that are made, and then those pieces have to be processed to make one continuous piece of DNA. So this is the slower side. And the fragments, these fragments, are referred to as Okazaki fragments. They're named after the researcher who first described them. And they have to be connected later, because you can't have a chromosome replicating in little pieces. The DNA has to all be connected to make, two complete, now double helical DNAs. And so this basically just describes what I've already said. The leading strand only needs one primer per, sort of replication bubble that opens up. We'll be talking about that terminology in a moment. But a lagging strand needs new primers around every 1 to 2. KB is thousands of bases. So every 1,000 to 2,000 nucleotides, there's going to be a new RNA primer made and a new piece of DNA made. And this is sort of what it looks like, probably much neater and nicer than what I showed. So notice the DNA molecules, notice the 3' and 5', they're super important. When the RNA primers are made, they are anti-parallel, just like DNA strands are. So if this is the 5' end of the DNA, the 5' end of the RNA primer is here, and the 3' end of it is here, and then the DNA is added, nucleotides are added onto here. And so this is the leading strand, and this just keeps adding, as the process keeps going, as the DNA opens, you can keep synthesizing. But as I said, the Okazaki fragments are generated on the lagging strand sides. And these are between 1 and 2,000 in size. And each Okazaki fragment is mostly DNA, but it has a little piece of RNA primer here. Maybe, maybe 6 to about 10 to 15 nucleotides long. They're not huge primers. You just need a little bit of RNA, and the RNA is complementary to the DNA, so it lines up in the correct place, and then the DNA is extended. So as I said, we don't want the actual pieces anymore, the Okazaki fragments. Once we have proceeded along, DNA polymerase 1, that is the first one that was discovered. DNA polymerase 3 is the one. Let's go back. DNA polymerase 3 is the one that's doing the majority of the replication work. All these new DNA strands that are made by POL3. But then DNA polymerase 1, what it will do is, if you have 2 Okazaki fragments, so that's the 5' end of it, and the 3' end of it, the polymerase actually binds to the end, the 3' end here, and starts adding DNA nucleotides. And as it moves along, it gets rid of, chews away the RNA nucleotides that are ahead of it, and then chews into this piece and replaces all the RNA with DNA. And then it leaves the little gap, which I will just... So what you end up with is, you end up with losing the RNA pieces that were here and generating just a tiny gap between these two pieces. And then there's an enzyme called DNA ligase that glues them together. Now, in molecular courses, you'll learn more about the exact way it happens. There's a little bit of complexity to how it actually works. But if you just remember that the two pieces, the 3' OH of this piece is connected to the 5' phosphate of this piece, and the little gap is sealed. And that DNA ligase is the enzyme that does that. And basically ATP, the energy to do the ligation, comes essentially from ATP. And as I said, there's additional steps and a little more complexity in here, but for our course, this is all you need to know at this point. As you go through additional courses, you will build your understanding further by adding more details. So this is how it works. On the DNA, to be replicated, there is what is referred to as an origin of replication. So that is special sequence, that DNA factors that are going to be used to open up the DNA to allow replication to occur. So separating the 2 original strands, which are these blue ones here, and they open up what is called a replication bubble. And it really looks like, if you were to look under like an electron microscope or something like that, you can see what looks like little bubbles along the DNA. This opens up more in each of the 2 directions. So it opens in both directions. So the kinds of images that you're usually going to be shown in both textbooks and in a lot of my lectures will just show you 1/2 of it. But it's good to keep in mind that although we might be talking about what's going on this side, that there will be, in the actual organism, there will be 2 of these. This is the replication fork. This is the replication fork, this is the replication fork, and this is the replication bubble. And then if you'll notice, this DNA, this blue one, goes 5' to 3' here. And then as this is the place where the DNA first opened up, so this is where the primer was made, here and here. And these are the leading strands. So the strand on the top is the leading strand in this direction. but the leading strand is on the bottom in the other direction. And this is the kind of thing you might have to draw for yourself more than one time to really kind of get it, to understand it. And then as the replication fork opens more, then we make the lagging strand primers, and we extend them and extend them, and then the next it opens a little bit more, and then we get this and this, and then it opens a little bit more, and we get this and this, and so on. So it opens up in this kind of sequential way. And it depends, like in a lot of eukaryotic chromosomes, there might be multiple origins of replication all along the chromosomes. In a bacterium or something like that, there might just be one origin of replication in the circular chromosome, and it extends all the way around until the replication forks kind of meet each other, and now you have two circular DNAs instead of one. Okay, and so yeah, I animated this. I'll show this in class as well too. So in eukaryotic chromosomes, as I said, there might be multiple origins of replication, and they start, in these very precise places with certain proteins that open up the DNA and start unwinding it, helicases, for example. That do some unwinding and opening up so that the DNA is available to be replicated. And then these all extend away, so that the replication forks in each replication bubble extend away from each other, and they get closer and closer as replication proceeds until you get to the end of the process when all of them kind of join up, and you now have your two complete daughter chromosomes or daughter molecules. Yeah, and so having more DNA doesn't necessarily mean you will take longer to replicate it, because multiple places open up and start replicating all at the same time. Simultaneously means at the same time, across multiple locations on the chromosomes. Okay, so that's just a really brief introduction to the whole process. And we'll go through in a little bit more detail in the class, just hammering away at some of the key points. So I'll be asking you questions about, the basic DNA structure. We'll be going through those experiments that I talked about, and so on. And this is the key concept that we're working through in the next few lectures. So we're going to talk about sort of DNA, the structure of it, how it gets replicated, and nucleotides and so on, what they are. Then we'll talk about the transcription process and how the RNA is made, and how it, in eukaryotes, the processing that takes place, and so on. Then it gets sent out of the nucleus in the case of eukaryotes to get translated into protein, and we'll talk about several subjects here: the genetic code, how it works, how it was discovered and understood. The discovery is quite interesting because there's a lot of very few tools and lots of thinking and figuring out. That's pretty impressive when you consider, the kinds of tools that people had to work with at that time. A little bit about mutations and what makes a mutation, and how a change in DNA sequence alters a protein, and then what that would mean in terms of, the phenotypic changes, and then just the steps of translation. So we'll spend a little bit of time on here. And we'll briefly mention that not all RNAs that are made are going to be translated into protein. There are some, for example, ribosomal RNA, that are part of ribosomes that do the actual translation part, and then transfer RNAs, tRNAs, that are also take part in the translation process. And there's other functional RNAs that we may touch on a little bit. And so by the time we get through to about the end of lecture 7 or so, then we're going to, I might have a separate lecture here on mutations specifically. I'll be working on that plan as we proceed through the course. So most of what I've decided pertains only to the first few the first couple of weeks, and then as we proceed, I'll see how things are going with the class, and then I'll tweak some of the topics and so on to suit the class. And I think that's all that you need to know at this point. But this is the first in a series of topics that's going to take us through this general overall principle of genetics.